Next up, we have Dr. Roxana Myers. She is the research nematologist at the USDA's Daniel K. Inouye U.S. Pacific Basin Agricultural Research Center, where her responsibilities focus on the development of cost-effective and sustainable approaches to the control of plant parasitic nematodes. Her areas of research include evaluating methods of media disinfestation, testing the efficacy of nematicides in Hawaii's agricultural systems, utilizing predatory nematodes and entomopathogenic nematodes as biological control agents, and developing crop plants with resistance or tolerance to plant parasitic nematodes. Her presentation today is entitled Plant Parasitic Nematodes in Ornamental Crops. Dr. Myers, thank you for joining us today and being patient.
And good afternoon, everybody. And thank you for the Hawaii Floral and Nursery Association for inviting me to speak today and give you an update on my research of nematology. I'll be discussing plant parasitic nematode management and ornamental production. And I'm a research nematologist at the USDA ARS Daniel K. Inouye U.S. Pacific Basin Agriculture Research Center. And thank you for having me here today. The subject of my talk will focus on the burrowing nematode, Rhodophilus simelis, which is a microscopic non-segmented roundworm. It's a migratory endoparasite, which means it spends most of its life within the plant itself, usually in the root system, but it can also be found in the stems of some plants, including anthurium. This means that it can be disseminated through cuttings, divisions, and vegetative propagation. And these videos here are pictures of our cultures that we have inside the lab of burrowing nematode, because we need to raise them in order to experiment on them and learn more about these organisms. So how do nematodes damage crops? Well, they destroy and weaken the root system, which interferes with the uptake of water and nutrients. They cause stunting of the plant, and this weakens the plant's immune system. All of this results in poor crop yields. And this photo is a picture of some anthurium roots at the beginning of a burrowing nematode infestation. You can see there's some brown necrotic spots, which eventually lead into a devastating root rot. So there is an integrated pest management plan for controlling burrowing nematodes, and this involves prevention, eradication, post-resistance, and curative control. And today we'll be focusing on the last two, and we'll start off discussing curative control methods. So I'd like to share with you an experiment that was conducted at the University of Hawaii Extension Agency at the Waikea Experiment Station, where we'll be visiting tomorrow on our field trip. This was done in collaboration with Brian Bush, Joanne Lichty, Arnold Hara, and Brent Sipes of the University of Hawaii CTAHR. So our goal was to evaluate a number of nematicides against burrowing nematode and see if we could control this pest. So this was done in a nematicide box trial, which is a little bit different than the trials that we have been doing with the growers. And this was to simulate a field trial. So we did grow the anthuriums in cinder, but we had them contained into separate boxes. And I was able to do final whole plant destructive assays at the end. Usually we don't like to take all the growers' plants out of their field. So this gives me an option to look at a different analysis. So what we did was we inoculated each of the cinder boxes with burrowing nematodes, 2,000 juveniles per plant, and there was 10 plants in each box. And this was replicated 4 times. We did nematode sampling of cinder and roots quarterly. And at the end, we did a final whole plant assay where we removed half of the plants, 5 from each box. And we took the root weights and we did root health ratings where we evaluated the percentage of root rot. And this is the cultivar that we tested, Toyama Peach. And thank you to our grower collaborators who provided the plant material. So in this trial, we decided to test 4 different chemical nematicides, 3 chemical nematicides and 1 biological nematicide. The biological is called Venere, also known as Magistine, and it's a Burkholderia species. And the 3 chemicals we tested was Broadform, which is the ornamental label. And there's also another label called Luna Sensation, which we've tested in the past. And this is Fluopyrium plus the fungicide trifloxystrobin. We also tested vellum, also known as indemnify, whose active ingredient is fluopyram. And in addition, we tested nimids, which is fluorosulfone. And this was the first time I had ever worked with nimids. You know, for the last 4 years, we've been doing quite a bit of tests with the fluopyram, which has been very promising. So we applied these treatments every 3 months at the maximum allowable label weight. And this is what the trial looked like in the boxes. And I have to tell you that Toyama peach is a good grower, and I think it could be quite tolerant to the nematodes. So if you ever want to grow that one, we had no problem growing it very well. And I think the nematodes liked it too, but they didn't seem to put on a lot of visible foliar damage symptoms. So this is a picture you can see in the middle of kind of what the boxes look like. We try to keep them contained, at least the plant, the under parts of the plant, separate from each other. So this was the result of the final assay where I removed the entire root system, and we compiled those 5 numbers into one big composite sample, and we took 200 grams out of that, and then we processed that to extract the burrowing nematodes. And so one thing you can tell right off the bat is that all the little A's mean they're not statistically different. So even though there's, some bars look higher than others, when you go to analyzing it using statistics, they're pretty much all the same. But in general, the vellum and nimids counts were very low at the end of the trial inside the root compared to the water. So over time, we had been doing this quarterly sampling. And so this is what the quarterly sampling looked like over the 12-month period. And these were burrowing nematodes extracted from the roots over that time. And these also were not statistically significant. And we did see that the vellum 1 had the least number of nematodes compared to the water and the other treatments. And when we looked at the nematodes in the cinder outside of the root system, vellum 1 also had the lower numbers, as did the nimids. However, as I said, they're not statistically significant, and I found in a number of studies, counting nematodes isn't really the best way to evaluate a product or evaluate a plant's tolerance to the nematodes. So we like to look at other things like the root weights, which I thought gave a really nice example of how these products performed. And you can see if you look at the little letters above the bars, if they're different, that means they are statistically different, and then they are important. So the vellum had the highest root weight of all the different treatments, followed by broad form. And the nimids, venerate, and water, all the root weights were very similar and about the same. So then we looked at a root health rating on a 1 to 5 scale based on root rot. 1 being like the uninoculated, no nematodes, nice, fresh, white root system. And a 4 or 5 would be untreated, extreme root rot, inoculated with the nematodes, showing a lot of visible nematode damage. And so this also followed the same trend, except in reverse, because a high number means that there's more nematode damage. So the low number means the roots looked healthier. So the healthiest roots were from the vellum treated, which did look the best and were significantly different than the other treatments. Broad form came in second with a mediocre root rot rating, and the vellum, the venerate, nimids, and water were all pretty similar. At least in this one, the water did come out at the top, which you would expect to be the most damaging, with no chemical or biological treatments. So just to conclude with that study, the nematode populations weren't greatly reduced by any of the nematicide tested. There wasn't really any silver bullet you could go in and apply and they would all go away. But we did see nice, robust root development, less root rot, and improved plant vigor in the fluopyram treated plots, which were the vellum and the broad form. Fluopyram appears to have potential in mitigating the damage caused by burrowing nematode in Anthurium cut flower production. And we've seen this before. I think this kind of follows up all the other studies that we've done where you did see a lot of nice healthy roots and improved plant vigor with these applications. So here's the broad form ornamental label. So this is approved for use in Hawaii. And unfortunately, right now, the vellum, which is the Floriculture only, does not have an ornamental label for Hawaii just yet. But hopefully that's in the works soon. I think the reason the vellum worked more effectively was because the active ingredient of the Floriculture is in a higher amount, and you can apply it with a higher application rate. So that's probably why we got a little bit of difference there, and that was a little bit improved. But Floriculture definitely seems to have some potential. So one thing to remember if you're going to use it is to apply at the first site of a burrowing nematode infestation. You should target the application to the root system under the leaf canopy, so you can use drop nozzles. And what we've seen in the experiment before this one was that plants with severe damage from nematode infestation won't recover as fast with chemical treatments. So if you start to notice any signs, you need to start your treatment procedures right away. Okay, so now I'm going to move into host plant resistance. This is a very exciting project that we're doing in collaboration with Paulo Vieira and Kathy Camo, who we're working at the U.S. National Arboretum. Catherine has retired, and Paulo just got a new position at the nematology lab at Beltsville at USDA ARS, and we're very excited about that and to have him permanently join the ARS family. And luckily, he still wants to continue collaborating with us because he thinks burrowing nematodes are super interesting. And so you'll see why when you see some of the research that he's done. So we'll be talking about quickly about the transcriptome, but more importantly about the new genes that are being developed for host plant resistance. So a few years back, we did have the burrowing nematode transcriptome sequence. We started out just an overall reference transcriptome, and then we wanted to move and just do a special gland cell transcriptome. So the esophageal gland cells from the nematode were microaspirated, and then you could collect the proteins inside of there and have those sequenced. So our focus was on the secreted proteins, and what these are the proteins that help the nematode to be able to infect the plant and to feed and complete its reproduction off the plant. So you can see if there is some way that we could stop the production of these proteins, we could possibly develop resistance. So we started off with a list of 41 potential effectors that we wanted to look at. And so some of these proteins, what they do is they can break down the cell wall so the nematode can feed on them better. They can help the nematode uptake the cell wall content, the cell contents inside, and that's their nutrient source. And also they can actually try to block the plant's defense system so that they were able to more effectively parasitize the plant. So the first step was we wanted to look at the transcriptome, tried to pull out these gland cell expressed genes, and then we look at what other nematode genes have been discovered that are the same in other type of nematodes. So all of those initials are different nematode species that have been sequenced. And so we compared those, and there are different types, migratory and sedentary parasites. And then the one on the very end, CE, everybody's probably heard of, C. elegans is a free living nematode. So there's a lot of sequencing data we can compare. So there were like the basic ones that we found that were, the same in these other nematode species. But most excitingly was this whole list of gland cell genes that have never been discovered before. And so either they're, they're probably very specific to the burrowing nematode, which is great if we want to just target that nematode species. And especially it won't affect any, if your control method won't affect any free living beneficial nematodes, which is nice. We didn't see it in C. elegans. So if they haven't found it in there, it doesn't exist. So this has been great. We found many new genes that no one's ever heard of before. And this is all going into a paper which has been accepted for publication in PLOS Pathogens with Paulo as the primary author. And it's a very thorough and interesting paper if anybody's interested in digging into this further. So when you find these genes, on the computer, you have the gene sequences, that's one thing. But now we need to know, were they just some kind of contamination? How come there's all these new genes? We have to confirm that these genes actually do exist in the nematode. So what you can do is this procedure called in situ hybridization, where you can target those proteins inside of the nematode and then have them like fluoresce. So you see these dark areas within the esophageal glands, that's where those proteins are made, and those are getting bound, the mRNA is getting bound up. And so that's why we're able to view them. So this is one great confirmation method, and he did hundreds of these, and definitely those proteins are in there. So that's good news. We can move forward to the next step. And the next step would be to transform like carrot hairy roots, and then introduce the burrowing nematodes into them and see if they're able to feed and uptake these constructs meant to block these proteins. So at the top, you see the different stages. On the left is day 1 after inoculation, the nematodes starting to penetrate the root. By day 3, they're fully inside the root system. Day 7 and 30 days, you can see they've already reproduced, and they're very happy 30 days after inoculation in the controls. However, in the ones with the very interesting gene constructs, we did see very different results. So what he did here was he wanted to make sure that these genes were being expressed by the plant, and that's what those red bars are. The darker the red, the more expression was in the plant. And what's great is you see all these pioneer genes right up at the top with the highest expression. So those are the best candidates for being, excellent sources of resistance. And then on the left, you can see as the nematode uptakes it, on many of them, the expression gets a little bit higher within the nematode. So that was some really great, important work. And so the next step would be to evaluate these genes actually against burrowing nematode reproduction and see if they're gonna, stop these guys from completing their life cycle and making more and more nematodes. So these double-stranded RNA constructs were developed to silence these genes. We looked at movement genes as well as these very interesting effector genes. They were transformed into carrot hairy roots. And then we did the screening at our lab at PBARC to see how the burrowing nematodes would react. And then if we find anything successful, our ultimate goal here for this project is to put these genes into Anthurium. And so we can have some resistant Anthurium plants where you don't have to worry about this chemical control and all these other control methods. You can just follow your normal growing practices. So here's what the carrot hairy roots look like. They're just on these little plates in the lab, which allows us to do a lot of numbers. There was many lines that were transformed, and so each line will behave differently. But all of the lines allow us to, evaluate these different gene constructs over the long term. So we put 1,000 sterile burrowing nematodes onto each one of those plates, and we had many, many reps of each. And we just had boxes and boxes of these things, but it was so exciting to see the results. This is the fastest nematode assay I've ever done in 2 months. Typically, I have potted plant assays or field assays, which we run for at least a year at a time. So getting the results quickly was a lot of fun for me. And I could also watch the progress under the microscope. You can see in these top pictures, you can see the nematodes coming out. So when you see that, you know there's a lot of reproduction occurring. And I didn't really want to see a lot of reproduction occurring, but at least I know, the assay is working. However, not all of the plates had a lot of reproduction. Fortunately, 4 of the 6 constructs tested effectively reduced burrowing nematode reproduction. That was great. And it was more than we had even expected. And some of the most promising lines supported 400 times less nematodes than untransformed roots after this 2-month period. And what that means is I got less nematodes off of those plates than I put on them 2 months prior. And that is great news. And that's just what we want to see when we develop resistant Anthurium plants. So the future work is going to involve more screening of these lines that we have. Also, we're going to get some new lines. We want to test some more of these new effector genes, especially those with had a lot of expression and look like they might have a lot of potential. And as soon as we decide on a handful of the best ones, we are going to make constructs for anthurium transformation and start transforming anthurium so we can get those into the pipeline with the rest of the great characteristics that we're working on at PBARC. So in conclusion, I just wanted to go over one last time the IPM plan for managing burrowing nematodes, just to keep it fresh in everybody's mind that there are steps that you can take to try to mitigate the damage done by this pest. The first one, of course, is prevention. Start clean and stay clean. You know, use clean plant material, watch your water and worker movement. That's crucial. You don't want to contaminate any clean areas of your farm. The second one, if you do get the nematode, try to eradicate them. You can do this using hot water baths to disinfect cuttings. And that's been adopted by some growers with very good success. You can also steam sterilize your cinder beds. You can either get newly fresh steams or you can steam the beds in the ground using a rubber tarp to force the steam down, and you can eradicate the nematodes in that way. Our ultimate goal, of course, is to develop host plant resistance, plants with resistance or tolerance to nematodes. That's always the most environmentally friendly and easiest adoptable approach to controlling nematodes. And finally, there is a curative control with fluopyram applications. You can reduce the nematodes in the cinder and also improve the plant vigor from the infested plants by using these chemicals. So I'd like to thank the members of my nematology lab, Kathy Mello, and our former student, Kimberly Escobar, our UH collaborators, and also our USDA collaborators. Thank you so much for moving this project forward to help out with the growers. And Glenn, is there any questions that you may have for me?
No, we haven't had any questions come in for you. However, I'd like to thank you for reviewing that IPM strategy. I'm sure it's of a benefit to the producers, and we look forward to that exciting work on the possibility of reducing the need for chemical control or reducing the amount of chemicals needed to produce healthy anthurium plants. So thank you very much, and we look forward to updates on your research. Thank you.
Well, I appreciate it. Thank you very much.
You're welcome.